Good morning. Today I'll be discussing a scientific report carried out in Africa. Contact with domestic dogs increases pathogen exposure in endangered African wild dogs. This report summarises a study carried out from 2001 to 2009, which was intended to find the rate of pathogen exposure in endangered African wild dogs by testing and analysing the blood samples of both African wild dogs and domestic dogs. The blood samples were tested and retrieved from 90 wild dogs in 19 different packs and 184 domestic dogs, which had absolutely no evidence of ever being vaccinated against any pathogen. The dogs were contained from two main land types: privately owned commercial ranches and community lands. They sorted from these two land types because both domestic dogs and wild dogs seek their homes in both these areas. Once the wild dogs were immobilised, blood samples were collected from the jugular vein, and the same process was then repeated for the domestic dogs, although they were not restrained and the blood was drawn from the cephalic vein. The blood samples were then stored and each was tested for a variety of different pathogens. Different techniques were used for sampling, from the rapid fluorescent focus inhibition test for the rabies virus to the serum neutralisation test to test the canine distemper virus. The results show strong evidence to support the claim that domestic dogs were the cause to the decreasing number of African wild dogs, even though antibodies against canine pathogens were found in both domestic dogs and wild dogs. Wild dogs that come in contact with the infected domestic dogs are at greater risk of catching the canine parvovirus, which 25% were in fact infected with, the rabies virus, 15% were infected with, the canine distemper virus, which 16% had been infected, and 4 other pathogens with diverse transmission mechanisms. Although these results aren't high, it will only take one wild dog to infect their whole pack. These viruses and pathogens are transmitted through infected feces, tick vectors, infected prey, as well as from mother to pup through the placenta, meaning the smaller the pack size, the more vulnerable they were to being infected. It appeared that although pathogens were found in the blood samples of wild dogs, if the dog appeared to be healthier, it was less likely that it would die from the pathogens it was infected with. This, however, was not the case for the domestic dogs, seeing as the numbers rapidly decreased once infected with the pathogens. Furthermore, wild dogs live in a small density pack, which means the chance of them actually coming into contact with an infected domestic dog is slim. However, if the interaction does occur, it does mean that it is more probable that the entire pack will become infected. The analysis of blood samples taken from both wild African dogs and domestic dogs in this specific report conclude that domestic dogs are the sole purpose and cause of transmitting canine pathogens to packs of wild dogs, thus resulting in a slow extinction rate and diminishing numbers of the African wild dog. Thank you.